Hello, my name is Jessica Moran and I am a PhD candidate with the CRC for honeybee products. Today I'm going to be talking about my research on developing a beehive breathalyser for American foulbrood, specifically on the volatile biomarkers that we've found and how they can be used for non-invasive diagnosis. Before I get to my talk, I thought I'd give you a quick introduction into who I am. So I grew up on a cattle farm, but I've always been a massive science nerd. I ended up going to the University of Western Australia to study a Bachelor of Science. And in 2014, I came across the Centre for Integrative Bee Research, and I began helping out with lab and field experiments. Professor Boris Baer liked to say that if you look too closely at honeybees, you get addicted. And that's exactly what happened to me. As soon as I opened the beehive, I was hooked. I ended up doing my honours with Boris on sperm motility in honeybees. But I was really excited when the CRC for honeybee products started, and I immediately joined Dr. Julia Grassel's Honeybee Health Research Group in 2018 to start my PhD. This is when I began studying American foulbrood. Since then, we have gone on to patent our biomarkers for AFB and secure extra funding from AgriFutures Australia to begin developing a beehive breathalyser, which is what I'll be talking about today. To start off with, what is American foulbrood, or as we tend to call it, AFB? AFB is a bacterial disease that can start with a single diseased larva and lead to a dead colony. The infection starts when the young larva is fed spores of the bacterium Panabacillus larvae. As the larva grows, the bacteria germinate and multiply in its gut. Eventually, the bacteria then break through the lining of the gut, and this causes a blood infection which kills the brood. It can be a larva or a pupa at this stage, and that depends on the genotype of the bacteria. The bacteria continue eating the brood from the inside out, and it turns it into this gooey, sticky mass that stinks. Eventually, this dries out into a crusty scale that contains billions of spores. Just one larva is turned into 2.5 billion spores, and it only takes less than 10 spores to cause a fatal infection in another larva. So as these spores move around the hive, more and more brood become infected and the honey is contaminated. This can spread to other hives through bees drifting or robbing, or beekeepers moving material around. And this is what AFB actually looks like. On the left are dead brood that have turned coffee coloured and are slumping in their cells. And on the right, this is an example of a rope test. If you probe AFB brood with a matchstick, then it will make a long rope between the cell and the stick. If we have a look at AFB across Australia, it's not a good story. AFB is present in all Australian states and territories. In mainland Australia, we have the policy that if you find AFB in your hive, you are required to destroy that colony. We do not allow the use of antibiotics for controlling AFB. This makes AFB a very expensive disease. An outbreak that destroyed 200 hives cost the beekeeper $1.5 million. This cost includes replacing colonies, but also the lost opportunity for honey production and being unable to fulfill pollination contracts. Based on a survey from 2006, 56% of beekeepers had a history of AFB in their apiary. So it's quite likely that as a beekeeper, you will experience an outbreak of AFB at some stage. That same survey also indicated that AFB is on the rise, with the number of AFB outbreaks increasing fivefold between 1971 and 2006. And AFB is continuing to be a problem, particularly as we increase pollination services that see beekeepers and hives coming together from all over Australia, but only about 10% of them are being checked for AFB. The reason only 10% of hives are inspected for AFB is due to the time and labour that it takes. Diagnosing AFB requires someone skilled at finding AFB to open the hive and check the brood for symptoms. So this makes diagnosis really difficult when beekeepers are running large numbers of hives or inexperienced with AFB, particularly if it's in an early stage of the outbreak where there's only a few infected brood cells to possibly find. So this is why we really want to develop a beehive breathalyser, a device that can detect and diagnose AFB from its smell in the beehive air without a beekeeper needing to open the hive. But to do this, we need to know what American foulbrood smells like. And this is because all smells are just chemical compounds in the air. When we smell a beehive, we are actually smelling the compounds that are given off by things in the hive. For example, from the honey, we could be smelling things like eucalyptol in the eucalyptus nectar. The hive material can also release a range of odours, such as pinene from the wooden boxes. And the bees themselves also release a range of compounds and pheromones, including this one here, which is beta-cis-ocimene, a brood pheromone. So if two hives smell different, they must have a different combination or abundance of compounds. We know that AFB hives smell different. They have that foul smell, and sniffer dogs are quite good at detecting outbreaks. But we needed to know what the exact chemical compounds were that caused that smell. And we can analyse the compounds with a gas chromatography mass spectrometer. Together with experts Micah Boland and Gavin Flametti, we used this very expensive GCMS to look at the compounds in the air around diseased and non-diseased larvae. The AFB brood that I analysed were generously donated by our bee biosecurity officers in WA and some anonymous beekeepers. The data that we get from GCMS looks like this. Each peak is a different compound. The taller the peak, the more abundant the compound, and we can look up the identity of that compound in a database using its ions. This is a simplified mass spec readout of a non-diseased sample of brood. This collection of smell compounds is what we call a volatile profile. It's basically a signature or a fingerprint of that sample. And this one contains over 100 compounds. When we compare it to the mass spec reading of an AFB sample, we can see that there are a number of compounds that are only present in the AFB sample. But keep in mind that larvae are little sponges for smell, and a lot of these compounds are from the hive material or what they've been eating. So that's why we needed to look at lots of samples to make sure we weren't getting excited about random compounds. So if we plot the volatile profiles or fingerprints of the samples, so that's each of these points representing the abundance of each of the 100 compounds, we can very quickly see differences between diseased American foulbrood samples and non-diseased samples of brood. So this was a really nice confirmation that we can tell the difference between AFB and non-diseased brood just from the chemical compounds. But we wanted to check that the volatile profile of AFB is distinct from other common causes of brood death. So I went back to the mass spec and collected some more data on brood that died from sac brood and brood that I had killed through either heat or cold shock and then let decompose for a day. Basically, I wanted to mimic what happens when we get chilled brood or when a hive overheats. What I found is that American foulbrood still has a very distinct volatile profile. It's quite different to sac brood, and basically all of the non-diseased samples, regardless of whether they were heat or cold treated, all gathered together. They were very similar to each other and very different from AFB. With just this data on volatile profiles, we were able to use a model that was able to classify the condition of the brood as being American foulbrood, sac brood, or non-diseased with an accuracy over 97%. At some point, I would also like to add European foulbrood to this figure, but WA doesn't have European foulbrood, so I'm still working on getting some samples that I can analyse. I dove into the analysis and found that it's only about 30 compounds that are the key to American foulbrood's volatile profile. They are what we call biomarkers. This is a heat map showing the abundance of these biomarker compounds in non-diseased and AFB diseased brood. Each row is a different volatile compound. Where it is dark, it's highly abundant, and where it's white, it's absent. So what you can see is that these biomarkers are either highly abundant or exclusive to American foulbrood. These are the compounds that if we could use a sensor to pick them up, then we would have a tool for detecting AFB. I should point out that we do know the identity of these compounds. We're just not making that information public until our patent is finalised. But all of the results that I've shown you were from lab experiments using larvae and pupae that we had removed from the hive. We still needed to make sure that we could detect these compounds in the hive. So with the help of Tiffany Bates, I did a field experiment at UWA using healthy and AFB colonies so I could analyse the compounds in the hive air. This helped us to further narrow down our list of biomarkers, those that occur in high enough concentrations to be detected in the hive. We were really excited that a number of our compounds are detectable and only present when AFB is in the hive. We were also able to see a link between the abundance of some of these compounds and the level of AFB. But what I found really exciting is that we can pick up AFB really early. Some of the biomarkers were detectable when there was less than 5 infected brood in the whole hive. This is a basic plot of the results we had of one of our compounds. This axis is the compound abundance, so a measure of the size of that peak in the mass spec reading. And this is the number of AFB brood in the hive. So you can see that the compound is absent in healthy hives, but as soon as there is just a handful of AFB cells, we can detect that compound. And as the infection gets worse, the compound increases, which I think is pretty cool. So now we are moving forward with our volatile biomarkers to create a portable sensor device that can diagnose AFB from beehive air, effectively a beehive breathalyser. And this needs to be a practical device that is both sensitive and accurate, giving fast results for beekeepers, while staying cost effective and user-friendly, and also robust. That is, it can handle a bit of honey on it or being bounced around in the truck. The trouble is we haven't found any existing sensor technology for our compounds that is able to tick these boxes. So we're going to develop our own sensors. AgriFutures Australia are investing over $500,000 in our sensor project, and Dr. Julia Grassel has assembled a team of sensor and chemistry experts from the University of Western Australia, plus me as the PhD student, to develop the sensors and then build and test a beehive breathalyser prototype. We're hoping to turn this into a commercial device within the next 5 years. So this brings me to the end of my talk. Summing up, we have identified the compounds released from AFB that are specific to the disease and detectable in hive air. This means we can now use a targeted approach for sensing AFB. We are now starting to develop these sensors for a beehive breathalyser device. We had always planned to make this into a handheld portable device, but it could also be incorporated into an in-hive remote monitoring system. We're quite excited because we think this device will be a game changer for beekeepers. Instead of having to spend time opening hives, diagnosing American foulbrood will be as simple as breathalyzing a beehive. Using our device, beekeepers and biosecurity officers will be able to accurately screen hives for the disease. This will prevent diseased hives from entering pollination sites, keeping bees healthy and beekeepers in business. If you want to stay updated with the project, then visit the CRC website, or if you have specific questions for myself or Julia, our email addresses are at the bottom of the slide. Thanks very much.